The name of my study is Task Demand Dependent, a Neural Representation of Odor Information in the Olfactory Bulb and Posterior Piriform Cortex. In this study, researchers wanted to understand the functional role of miftural and tufted cells in the olfactory bulb. These cells are important because they receive sensory input from neurons and transmit the information to higher areas such as the anterior olfactory nucleus and piriform cortex. These miftural and tufted cells, or M&T cells, transmit information specific to odor representation, learning, behavioral context, and task demands. So the purpose of this study was to greater understand when these M&T cells are used in odor discrimination and behavioral demand. And so the study conducted with male mice, and they were given the presence of 4 separate odor pairs, which are like a set of 2 odors. And the base task was just to lick a water delivery device. And if they licked the device, they received water as reinforcement. And so the beginning task was a go-go task, which meant the mice had to lick the water in the presence of an odor, and if they did so, they were rewarded with water. The second task was the go-no-go task, and when the mice then had to differentiate between the 2 odors and perform what they call an odor discrimination task, so they could only lick the water delivery device in the presence of the proper odor, and if they did so with the proper odor, they received water as reinforcement. And so what the researchers were looking at is when the mice had to differentiate between just licking the water device with any odor present, and then having to differentiate between the 2 odors, they found different activation of these M&T cells. And the activation was measured by calcium signals that were given to the mice prior to the experiment. And so the different activation levels represented the functional role of these M&T cells in odor discrimination and behavioral tasks relating to sensory odor input. And so they were kind of able to understand better what the olfactory bulb is used for in mitigating behavioral responses to odor and sensory input. The main key finding of this study was that when they switched the mice between what they called the go-go task and the no-go task, the M&T cells showed odor discrimination and divergence in the go-no-go task because the mice had to differentiate between the 2 odors, but this activation of M&T cells vanished when they went to the go-go task, and all they had to do was just detect the stimulus or presence of an odor. And so that was the large key finding of the result, was the ability of the M&T cells to either be activated or not activated, depending on the behavior required or demanded of the mice.